Good morning, sir. Good
Good
morning,
morning.
ma'am.
Good morning.
Ma'am.
Morning.
Good morning, sir. Good morning, sir.
Morning.
Good morning, Vanaja.
Good morning, ma'am. Good morning, sir. Mahender, sir.
Good morning. Good morning, Dr. Gonzaga. Good morning, Dr. Shubhanagar. How
Thank
are
you,
you?
sir. I'm good, sir. It's past midnight for you, sir.
Oh yeah, just
Yeah,
past midnight.
yeah,
And I
yeah.
just had my coffee to keep myself active.
I know, I
I'm
know.
all set.
Yeah, yeah, I know. The time difference between India and US, almost 10 and a half hours. So either we have to adjust to your night time or you have to adjust to our daytime, something
I
like
know.
that.
I mean,
Good morning, madam. Good
Good
morning,
morning,
all.
Ishwari. Good morning,
Morning,
Ishwari. You're not to be seen, Ishwari. Please
madam.
switch on your video. Your video.
Oh, yeah, yeah.
Yeah. Good
Good morning,
morning.
madam.
Good
Good
morning.
morning, Subbarao, sir. Good morning, Vanaja.
Good morning. Good morning, Ishwari.
hi. Good morning, Ishwari. How are you?
Fine, fine. Fine, I am fine.
Incoming.
Just we'll wait for a few minutes, sir. 3 or 4 minutes, so that people will
Sure,
join.
sure.
So, Baro, are we
Subbarao
live?
garu,
Pardon?
recording permission ఇస్తారా దీన్ని
Ah,
వీడియోని?
it's sir. You can
Yes,
take.
sir. Good, good.
Thank you.
Yes, sir.
I'm just requesting in the Zoom
Yeah,
right
yeah,
now.
no problem.
So, first
I
of
think
all, I
WhatsApp
thank
లో
you.
మెసేజెస్ పెడుతున్నారు వాళ్ళు యూట్యూబ్ వీడియో ఇస్ నాట్ యాక్టివ్ అని.
No, sir. It is going live, sir. Just
Oh,
I'm on the
sorry.
YouTube itself. Just
Oh,
I'm
okay.
refreshed once. That's all.
I see.
Yes. already people started joining, sir.
సార్ సుబ్బారావు సార్ సుబ్బారావు సార్
వీడియో
మన వాళ్ళు లింక్ ఓపెన్ అవుతలేదు అంటుర్రు ఒక్కసారి
అవును
మళ్ళీ రీసెంట్
వీడియో నాట్
టీమ్ అని
అవైలబుల్ అని
పెట్టండి
వస్తుంది.
సార్
లేదమ్మా,
టీమ్ అని తల
జస్ట్ ఒకసారి రిఫ్రెష్ చేయమని చెప్పండి అమ్మా.
అవ్వట్లేదండి, ఐఎమ్ డూయింగ్ ఇట్. నో, వీడియో ఈజ్ నాట్ అవైలబుల్ అని వస్తుంది.
వీడియో ఇస్ నాట్ అవైలబుల్ అని వస్తుందా?
యా,
1 minute, sir. Let me check. Let me check, sir.
కొత్త లింక్ వచ్చింది, పాత లింక్ మాత్రం రావట్లేదు ఫర్ సమ్ రీజన్. yes
1 minute, sir. I will try to do it. Hello.
యా, కొత్త లింక్ ఓపెన్ అయింది. ఐ థింక్ ద ఓల్డ్ లింక్ ఈజ్ నాట్ ఓపెనింగ్ ఫర్ సమ్ రీజన్. కొత్త లింక్ వచ్చింది, పాత లింక్ మాత్రం రావట్లేదు.
Hello. मुझे भी एक सर sir, new link has been posted, sir, to everybody.
ఓకే, ఐ థింక్ నేను కూడా పోస్ట్ చేయాలి. మా దే ఆర్ ఆల్ రెస్పాండింగ్ టు మీ.
2 minutes time, sir, no problem.
న్యూ లింక్ అందరికి పోస్ట్ చేయాలి కదా మరి.
వనజ గ్రూప్ లో కూడా పోస్ట్ చేయమ్మా.
ఓకే మ్యామ్, షూర్.
ఈశ్వరి, నీవు ఒక 2 minutes ఉండి మిగతా గ్రూప్స్ లో నెట్లో పోస్ట్ చేయమ్మా.
ఓకే సార్.
జస్ట్ యూ కెన్ మ్యూట్ యువర్ సెల్ఫ్ అండ్ డూ దట్ వర్క్ అమ్మా. వీడియో మ్యూట్.
ఓకే సార్. ఓకే.
Since how many years are you there, sir?
మా గ్రూప్ లో పెట్టా, రీసెర్చ్ గ్రూప్ లో.
సార్ నాకు ఇంకా రాలేదు, మీరు నాకు పంపించండి, నేను ఫార్వర్డ్ చేస్తాను.
Why did it happen? గ్రూప్ లో ఉంది చూడమ్మా ఈశ్వరి.
హలో వనజ,
గ్రూప్ లో ఉంది.
ఓకే సార్. థర్డ్ గ్రూప్ లో నాకు ఫార్వర్డ్ చేయడం వస్తలేదు సార్.
నేను చేశాను ఈశ్వరి.
ఓకే ఓకే, థ్యాంక్ యూ.
హలో. क्यों ऐसा हुआ है प्रभा? उसमें जो है ना वही चाहिए ना, वो जो मुझे वही। ठीक है। Sir, now it is properly working, sir. Already 73 members, participants have joined, sir. You are muted, sir.
ఓ సారీ, నాకు మెసేజెస్ వస్తున్నాయి. నేను ఫార్వర్డ్ చేసిన గ్రూప్స్ అందరూ కూడా మెసేజ్ ఫార్వర్డ్ క్విక్లీ ఇన్ 2 minutes.
We are doing it, sir. Already we have created a WhatsApp group, so single message will reach to everybody, sir.
Let's see. Just give me 1 second.
Otherwise, just we'll wait for another 2-3 minutes, sir. You can just try to forward the messages to those who are waiting.
All right, I'm all set.
మీకు 1:30 అయిపోతుందేమో కదా సార్ అయిపోయేసరికి మహేందర్ సార్.
Almost 1:30. That's okay, fine.
Thank you for accepting our invitation.
My pleasure.
Sir, how is your place, sir, there? So much of disturbance is there due to COVID.
The credit goes to Ishwari. She was like, you gotta do it. So, okay.
Thank you for obliging Ishwari, sir.
Thank you, Mahender.
సార్ మీ వాయిస్ కొద్దిగా
Clarity లేదా?
ఉంది, క్లారిటీ ఉంది, కొంచెం వాయిస్
హైలో మాట్లాడమంటారా?
కొంచెం ప్లీజ్.
Okay, sure.
Subbarao,
Shall we start, ma'am?
shall we start?
Sir, shall we start, sir? 81 members are there already participants. Ishwari, shall we start?
Since how many years you are there, sir?
Approximately 20 years.
Oh, so as soon as you completed your masters, you.
Not quite 20 years. I came in 2003.
Okay.
So 17 years.
Yeah. Long time.
Yeah.
సార్ ఇఫ్ యూ డోంట్ మైండ్ ఇంకొంచెం లౌడ్ గా చెప్పాలి సార్.
Oh no, sorry.
It's good.
How about now? Better?
ఎస్ సార్, అవును సార్, ఎఫెక్టివ్. థ్యాంక్ యూ. మేడం సార్ TV9 లో కూడా డిబేట్ లో
Participate చేశారా? Yeah.
పార్టిసిపేట్ చేశారు.
Yeah, a couple of weeks ago.
Yeah.
Yeah, shall we start, sir?
Sure. Go ahead.
Okay.
Yeah.
I don't know, sir. All the participants who have joined, thank you and good morning to everybody. And would like to start the session with the permission of our principal, ma'am, Dr. Shobana, ma'am.
Yes, please go ahead, Subbarao.
Yeah, and I welcome Dr. Mahender Dewal, who is who is working in America, but he is our Telugu Bidda and Telangana Bidda. And we know that it is very great privilege for us that our country people and our state people and Hyderabad is going to the crossing the borders and doing a great work. Sir, it is really a privilege for us to come here and participate in this webinar, sir. And I hope that your talk will give some hope to all of us regarding the COVID drugs, and especially to the young minds who are there in the seminar. They will listen to you and learn, and they may go to anywhere to achieve what you have done, sir. With this small preface, I would like our principal, ma'am, Dr. Shobana, ma'am, to welcome all the participants to this webinar. Ma'am,
Yeah.
please.
Yeah, sure. Thank you, Subbarao. Good morning to one and all. Good morning, or maybe it is past midnight, to some of the overseas participants who have kept themselves awake, including our guest speaker, to present this webinar to our students, our faculty, as well as a number of faculty members and students from different colleges in Hyderabad as well as all over the country. And it is indeed our proud privilege and honor, sir, to have you present the webinar for us. And we are extremely thankful and grateful to you for having accepted our invitation and having stayed awake for delivering this webinar past midnight. And I'm sure it's going to take 1 or 1.5 or 2 hours as such by the time you complete your presentation. So it will be almost early morning for you, sir. So I'm extremely grateful and thankful for that, sir. And it is Sarojini Naidu Vanita Mahavidyalaya, our institution, sir, where I'm sure, you being a Hyderabadi, you pretty well know about this institution, which is sponsored and managed by Exhibition Society and Usmania Graduates Association. This was started way back in 1962, sir. So it's almost 58 years of its existence, and n number of batches have passed, graduated, post-graduated through our institution, and many of them are well placed in India as well as well placed abroad. And your talk, I'm sure, is going to inspire our young minds here, where it is not only the talk on COVID, but it is also a presentation which will enlighten them about the research activities that are taking place all over the world. As we all know, pure science research is not much in India, so I'm sure your talk is going to inspire our young minds to take up research as their career. And you being from the prestigious place of Massachusetts, where it's a dream of every young mind to reach to Massachusetts, and your expansion technologies, which is an affiliate to MIT, it's indeed a great service that you are doing, sir, and I'm sure you will be an inspiration to all our young minds. So I welcome you, sir, to this webinar, and apologies for keeping you awake till late night, sir. So welcome to you, and as well as all the other overseas participants and dear participants in India. So let's have a great talk from a great man from Massachusetts. Thank you.
Thank you, ma'am. Thank you for your welcome words. And now I request Dr. Vanaja Reddy, Head Department of Chemistry.
Subbarao, you're muted.
Uh, yes. Th- Thank you, ma'am. Thank you for your welcome words. And now I request Dr. Vanaja Reddy, Head Department of Chemistry, Sarojini Naidu Vanta Mahavidyalaya, to introduce today's speaker, Dr. Mahender Dewal.
Thank you, Subbarao sir. Good morning, one and all. A warm welcome to all participants. We are extremely honored and privileged to have an internationally renowned among guest speaker, Dr. Mahender Dewal, founding member, Head of Research and Development, Expansion Technologies. He is going to enlighten us on the use of expansion microscopy of subcellular structures like coronavirus, its evolution towards COVID-19, various different virulent species, and underlying chemistry of COVID-19 from cause to cure. We are looking forward for hearing you and understand how to deal with the pandemic and mitigate its ill effects. Now, I am going to read out detailed biodata of Dr. Mahender Dewal. Fear, left unchecked, can spread like a virus. The first woman Nobel laureate and the only woman to win the Nobel Prize twice, a Polish chemist, Marie Curie, said, I quote, nothing in life is to be feared, it is only to be understood. I take this opportunity to introduce our speaker of the day, Dr. Mahender Dewal, a distinguished scientist in the field of bioorganic chemistry and chemical biology. He is one of the founding members of Expansion Technologies, a biotech startup at Cambridge, Massachusetts, USA. We are proud to state that he has his school and college education from Usmania University campus in Telangana and started his research career as Junior Research Fellow at CSIR, Indian Institute of Chemical Technology, Hyderabad. Then he moved to the University of South Carolina in USA for his PhD, where he successfully completed and accomplished synthesis and characterization of bisuria microcyclic supramolecular nanostructures. He completed his postdoctoral research from Department of Pharmaceutical Sciences, Wayne State University, where he worked on 3 projects. The first one, synthesis of protein-protein interaction inhibitors for the treatment of human cytomegalovirus infections. Second one, studied roles of amino acids present in the active site of N5-CAIR synthetase, a potential antibacterial target. Third one, synthesis and mechanism studies of membrane-targeted antibiotic for methicillin and vancomycin-resistant Staphylococcus aureus. Then he moved to MIT as a research scientist in chemical biology, where he studied N-glycosylation biosynthetic pathways and role of unfolded protein response in cancer. Also studied biosynthetic pathways that restrict HIV replication process. Presently, as principal scientist and head of research and development, he is leading a team of scientists working on early stages diagnosis of various cancers, breast, prostate, bladder, melanoma, renal, and skin diseases in pathological setup. He has 17 research publications to his credit. Two articles which highlights his work are published in Hot Article in Organic Biomolecular Chemistry, 5th August 2011. Another featured in Research Highlights in Nature Chemistry, 11th July 2008. He currently holds 4 US and international patents. Dr. Mahender is peer reviewer for European Journal of Organic Chemistry, European Journal of Medicinal Chemistry Research, and Tetrahedron. He received 2008 Graduate School Award and 2005 Cancer Research Department of Chemistry and Biochemistry. He has served as President and Advisory Board Member of Wayne State University Postdoctoral Association in 2011-12. Committee Member, Department of Pharmaceutical Sciences Strategic Plan 2012 to 16. He also served as Vice President of MIT Ventureship Club, which provides guidance and encourages early startup companies from MIT students and postdoctorals. Now, I cordially welcome to present his talk. Thank you.
Thank you, Vanaja, and thank you for your detailed presentation of biodata. It is a very rich biodata. And now I request Dr. Mahender Dewal garu to start his lecture.
Thank you so much, Dr. Vanaja garu, for such nice words about me. Good morning, everyone. Can you all hear me well?
Yes, sir. You can proceed.
All right. So let me set up my presentation first. Give me 1 second. And then I need to share this first. Yes. One second, bear with me.
Yeah, no problem.
I'm sharing the screen. Share the screen. Can you all see my presentation now?
Yes, sir. Yes, sir. We'll be able to.
All right. Good morning, everyone. First of all, I would like to thank Department of Chemistry, Sarojini Naidu, Vanita Mahavidyalaya, Principal Dr. Shobana Garu, and Dr. Ishwari, Dr. Vanaja Garu for inviting me to give this lecture today. I also would like to thank and appreciate Dr. Ishwari and Dr. Shobana, Mr. Subbarao Garu for all their hard work and efforts putting this event together. Thank you so much. Today, in my talk, first half, I'm going to talk about coronavirus evolution and infection mechanism, and also the components of the immune system fighting the coronavirus. In the second half, I'm going to talk about the treatment and vaccine options for the COVID-19, and also current progress in their development. COVID-19, a 6-letter word, is shaking the world from past 6 months. What is this? COVID-19 is an infectious disease caused by the most recently discovered novel coronavirus. As probably most of you are aware of that, this outbreak began in Wuhan province, China in December 2019. Quickly, within a few weeks, it spread out all over the world. Then the WHO, World Health Organization, declared it as a global pandemic. Let's take a look into the infection symptoms, COVID disease infection symptoms. It contains most common symptoms. Fever, it starts with basically most common symptoms: fever, dry cough, and tiredness. Recently, some data also suggests that some of the patients, approximately 65 to 75% of the patients, complained about losing the smell and taste. Now, most of the countries also included these two characteristics as identification or recognizing factors of the COVID-19 infections. As the virus takes over the body, the symptoms becomes severe and severe, which goes towards pneumonia, severe acute respiratory syndrome, SARS. That's why this coronavirus is also called SARS coronavirus, SARS-CoV, which leads to organ failure such as kidney failure, lungs failure, ultimately leading to the death. So when I said the COVID-19 infection caused by a recently discovered novel coronavirus, what is this makes this coronavirus novel? Why it is called novel? Is there any old coronaviruses already there? Let's take a look into the history a little bit. The coronaviruses are discovered in 1960s, the human coronaviruses. There are 4 generation coronavirus I have displayed here. All of them are discovered around 1960s. All of them are originated from either bats or rodents. However, they did not transmit straight from the bats or rodents to the humans. There were some intermediate hosts, such as camels, cows. But this first generation coronaviruses are not as pathogenic as the current SARS coronavirus. They were showed very mild symptoms like standard cold, common cold symptoms, and they're also infected mostly immunocompromised people, immunocompromised patients. However, in November 2002, in same Fushan province in China, another coronavirus epidemic started. This is called epidemic because this was spread only few countries, approximately 30 countries and 5 continents. And this is also originated from bats. But the intermediate host for this particular SARS coronavirus 1 are civet cats or raccoon dogs. The symptoms are SARS and pneumonia. This symptoms leads towards from mild symptoms to severe up to the death as well. Approximately 8,000 people all over the world, among those 30 countries, were infected by SARS coronavirus 1. 800 of them were dead. Same way, in 2012, also another coronavirus discovered, which is MERS coronavirus. It is discovered in Saudi Arabia. That's why it's called Middle East Respiratory Syndrome coronavirus. This was also declared by WHO as an epidemic, not a pandemic, because it was limited to certain regions, approximately 27 countries. Again, this one also originated from bats, but intermediate host is different, which is dromedary, a Somali camel. This virus is a little bit pathogenic, more pathogenic than the SARS coronavirus 1, which causes acute respiratory distress syndrome. Lethality is approximately 35%. 2,000 people from these countries were infected by this virus, and 700 people were dead. As you all know, in 2019 December, the SARS coronavirus 2 evolved. And as of now, I mean, again, this SARS coronavirus 2, current COVID-19 coronavirus, is also originated from bats, but the intermediate host is pangolins. As of an hour ago, I checked the lethality rate. Approximately 7.5 million people in all over the world impacted by this virus, which included 423,000 people who are dead so far. Having United States is the number 1 country in the infections, India is number 4. So far we have seen how this coronavirus evolved from coronavirus 1 via MERS coronavirus 2, SARS coronavirus 2. Let's take a look at the similarities and differences between these 3 pathogenic viruses compared to the four of the first generation viruses. All of these are originated from the bats. So the genome is 90 to 95% similar to the bat coronavirus genome. SARS coronavirus genome and SARS-2 coronavirus genome is approximately 80% similar to the SARS coronavirus 1 genome. They are like brothers, same family. And MERS coronavirus has a 50% different genomic information compared to the other two, SARS-1 and SARS-2. It is like a cousin of this family. And also another interesting difference if you see here is the receptor molecules where the virus lands on the human cell before the infection, which is ACE2 for SARS-1 and SARS-2, but it is different for MERS coronavirus. DPP4, which is dipeptidyl peptidase 4, is a receptor for MERS coronavirus. So, even though there are similarities between SARS-1 and SARS-2, SARS-2 is much more pathogenic and very highly transmissible. What are the reasons? What causing that? Let's take a look at that as well. The primary spread of SARS coronavirus 2 is from person to person is coughing, sneezing, speaking. Means people who speak loudly also the virus can come out of their mouth. And secondary spread of this virus is through air. It's an airborne virus. I mean, it's not. It's an air. Through air also it can transmit. And another source of the contamination is various different objects. It's very interesting. Take a look at this table here. This coronavirus 2 can be active up to 2 weeks at 4 degrees centigrade. It means in the fridge. But at the body temperature at 37 degrees centigrade, within 1 or 2 days, it's only active for that. In hot water, within 5 minutes, it will be cleared out. And take a look at the various different surfaces. Very interesting features this coronavirus has. Regular paper, tissue paper, 30 minutes to 3 hours. But a currency note and cardboard, those are also made of paper, but it survives up to 2 to 4 days. It's very surprising features, surprising characteristics this coronavirus has. An additional feature for this is also long incubation period. That means up to 2 weeks, an infected person does not show any symptoms and he may transmit the disease without knowing himself and people also. These are the reasons. That's why this pandemic is spreading like very virally. Now, what is the coronavirus? Let's take a look at the structural properties. Let's look a little bit more deep inside the genome and structural features of this coronavirus. Once Peter Medawar, he was a Nobel laureate, an immunologist, defined in general a virus as simply a piece of bad news wrapped in a protein. That's absolutely true. Like living cells, the viruses contain genome. It could be DNA, RNA. In coronavirus case, it is in positive sense RNA, which is securely wrapped by a unique nucleocapsid protein, and you can see that in here. And other couple of interesting features this coronavirus has is, first thing, these spike proteins. None of the RNA viruses belongs to this family has these spike features. Because of these spikes, it looks like crown. That's why this is called a coronavirus. And the second interesting feature is the spiral nature of the genome. The RNA. RNA is spirally, very well organizedly located inside the genome. That is also an unusual characteristic in compared with other RNA viruses. In general, viruses does not replicate. Basically, they're not considered as a living cells or living organisms. All the time they rely on the host cellular mechanisms, host cells. And the coronavirus, these viruses cannot replicate means they cannot become 1 to 2, 2 to 4, 4 to 16 or something like that. The mechanism is completely different that I'm going to talk about in coming slides. And coronavirus is also called zoonotic virus because zoonotic viruses are originally exist in the animals, but they can infect animals as well as humans. Now, when I said coronavirus genome is very well organized, let's take a look into the much more closer look into the genome. Another unique characteristic of this coronavirus is a large genome, which contains approximately 29,811 nucleotides, which encrypts encoding 29 proteins. This is the first virus has this large genome so far. Any other viruses, approximately in my knowledge, 20,000 nucleotides was the largest genome other viruses have. In this huge genome, it was divided into 2 parts. Let's look at closer look into what encrypted inside the genome. it divides into two pieces. Two parts. First part contains all the information related to the non-structural proteins. That means they're functionally active, not structurally. And rest of it is encodes structural proteins like spike protein, envelope protein, membrane protein, and nucleocapsid protein. Also, additionally, some accessory proteins that encrypts here. So, what kind of information we do have, structural or what kind of functional information we do have so far? Let's take a look at that. So, out of all the 16 non-structural proteins, out of 29, 16 are non-structural proteins, and 4 are structural proteins. The rest of the 9 are accessory proteins. So far, we are very well characterized only 3 non-structural proteins, 2 proteases and 1 RNA-dependent RNA polymerase. What proteases does is basically they disconnect the long polypeptide chains. So all the genome is translated as a single chain, and they have to be separated as individual proteins. Proteases, that's what proteases does. And this RNA-dependent RNA polymerase, which is also called replicase. This is one of the unique enzymes that SARS coronavirus has. This is a very smart enzyme which errorlessly transcribes the viral RNA into a negative strand, where the other cellular polymerases come in and make more and more RNA so that more virus can be produced. Structural proteins, the spike protein has its own unique characteristics, which basically is the primary reason for entering into the cell. It goes and binds to a receptor on the human cell, which is called angiotensin converting enzyme 2. Nucleocapsid protein, it securely protects the viral genome. And membrane and envelope proteins work together to give a nice secured structure, shape to the virus. And so far, the accessory proteins, there are 9 accessory proteins. The functions are still unknown. Only half of the information of this virus we know so far. Luckily, we know the genomic information. That is the most important part, and then that helps a lot in discovering the therapeutic agents and vaccines as well. Let's take a look at the cell entry mechanism. So, the spike protein, what it does is it attaches to the receptor, ACE2 receptor, and then slowly goes inside uses the cell membrane and creates an endosome. This process is called endocytosis. Once endosome is formed, again, then there's some functional conformational, structural conformational changes happens in the spike protein. It basically inserts itself into the cellular membrane and slowly releases the viral genomic RNA into the cytoplasmic of the cell, like coming inside out like this. And this cartoon picture shows basically the red color spike protein. It's a very large, approximately 1250 amino acid residues. And the blue one is the ACE2 receptor. Even though it's a large protein, this small functional part is very crucial for binding to the receptor, which is called receptor binding domain, RBD. Once the endocytosis process is completed and then viral RNA is released into the cytosol, first thing what happens is the cellular enzymes translates this positive sense RNA into the proteins, all the non-structural proteins and other structural proteins as well. First, the replicase will be released, the proteases will be released. As soon as the replicase released, then what it does, it immediately transcribes the positive sense RNA into a negative sense RNA, which becomes a template to make more and more RNA for the all the other entire genomes and other structural protein RNAs and accessory protein RNAs. Once these are produced, they will be translated and transported into other parts of the cell, like endoplasmic reticulum and Golgi apparatus, where they mature, they refine. And after that, once they're refined, RNA comes from the cytosol, from ribosome to one place, and the structural proteins come from from the Golgi apparatus. They assemble together to create a completely functional virus and a process called exocytosis. And similarly, opposite to the endocytosis, it will be released into the outside of the cell, and the virus is now ready. All this mechanism is basically like a assembling a car. For example, if you take Maruti Suzuki, the engine is made in Japan, the tires are made in somewhere in Mumbai, and the body is made somewhere in Kolkata. All of them were shipped to Ahmedabad, where the central assembly facility, the car will be assembled. That's how the viral. It won't replicate 1 to 2, 2 to 4 like that. And it completely utilizes, hijacks all the cellular mechanisms of the human cell. Once virus released, when the virus infected and once the virus released into the bloodstream, then the immune surveillance system, body surveillance system activated. Let's talk about the immune system. And the immune system is, there are two types of immune systems exist in the human body. First one is innate immunity, which comes with by birth. Everyone will have innate immunity by birth. And this is the frontline defense. It acts non-specifically. Within hours, few hours, less than 12 hours, it will be activated to fight with the infection that enters into the body. It's like a police force, basically, attached the virus. The second one is adaptive immunity, which will be developed by as we grow. Adaptive immunity doesn't come by birth. And this is like a backup force, like a reserved army, which will, when innate immunity is not able to control the viral infection, the adaptive immunity comes in place and start fighting with the virus. Both immunity systems contains various different cell types that I'm going to do a little more detailed explanation in upcoming slides. One important thing we need to keep in mind that this adaptive immunity can be strengthened by healthy diet and active lifestyle. And another thing is, since adaptive immunity is not present in the newborn babies, that's why they are kind of more prone to the infections, viral or any bacterial infections. And also, immunity system, all these cells, even though they exist in the older people, like above 60 or 70, but the functions kind of slows down. That's why they are also kind of immunocompromised. Let's talk about the innate immunity first. It has lots of various different cell types: basophils, monocytes, neutrophils, eosinophils, and so on. But I'm going to talk about most important cells: macrophages and nature killer cells and antigen presenting cells. When a virus infects the cell or human body, the very first thing immune system does is releasing some chemicals or protein molecules which are called cytokines and chemokines. What these molecules does is they just go and bind to the virus infected cells and viruses. They are kind of identifiers, kind of signaling molecules. When they bind to the infected cells and the virus, immediately macrophages attacks the infected virus where the identifiers are there, and immediately they catch it. It eats up the virus particles based on the identifiers. And then nature killer cells, immediately, these are kind of go to the virus infected cell and activates the suicide mechanisms to kill the infected cell so that the virus won't spread out more. As soon as it kills the virus infected cell, macrophages come and then clears it out by eating it not to make any poisonous molecules. While they are doing their job, the antigen presenting cells, what they do is they go and identifies the viral infections and then takes the signature molecules from the virus and runs towards the adaptive immunity system. Adaptive immunity system has basically two different types of cells: T cells and beta cells. When the antigen presenting cells brings them selective molecules, identification molecules from the virus, they give it to the T cells. T cells generates the two types of mechanisms. First, the T cell immediately finds that infected cell and virus, like a nature killer cell and macrophage, it kills, just lyses them and clears them out. And the second thing is they also generates memory cells, which remembers the characteristic features of the virus, and remembers how to fight this virus in future if comes and attacks. What and other type of cells while they are doing these job, beta cells generates antibodies which are called neutralizing antibodies. These neutralizing antibodies basically arrest the virus. They surround the virus and inactivates it. As soon as it is inactivated, the T cells or macrophages attacks the virus and clears them out from the body. However, this SARS coronavirus 2 is much powerful and smarter because somehow it escapes the immune system to that's why it's spreading very fast and rapidly. How does this coronavirus, SARS coronavirus 2, escapes the immune system? Now we don't know much mechanistic details, but there are some hypotheses that I'm going to explain you in a minute. The very first thing the virus does is it basically diverts the immune system by sending the wrong signals, which gives the immune system to produce lots and lots of cytokine chemokine molecules, which is called cytokine storm. These cytokine molecules are also two types: pro-inflammatory, anti-inflammatory. Anti-inflammatory are attacks on the viruses and virus infected cells. Pro-inflammatory attacks on the healthy cells as healthy cells. So when the large amount is produced, they don't know what to do. They start attacking the healthy human cells, the cytokines, the pro-inflammatory cytokines. They will go and bind to the healthy human cells. Then macrophages, T killer cells go and start killing those healthy human cells, which causes the tissue damage in the organ, which ultimately leads to the organ failure, and multiple organ failure leads to the death. This is how it's kind of escaping and invading the human body, the SARS-CoV-2 2. But how? There are some hypotheses. Some data suggests that accessory proteins in the SARS coronavirus 2 helps the virus to divert the immune system. The second thing is sugar coating on the SARS coronavirus spike protein. The spike protein heavily glycosylated, modified or decorated with the sugar molecules. Sometimes they also disguise the spike protein interaction sites, and that's how immune system may not be identifying the spike protein, so that successfully not killing the virus. The other possibility is this is the spike protein. As I mentioned in the previous slide, this is whole big protein, and this is the most important part, which is a RBD, receptor binding domain, right? This also exists in two conformations. Active conformation, which is this, which binds to the ACE2 receptor on the cell. But there is another conformation, which is, this is the active, but this is a passive conformation, which is kind of hiding, which doesn't bind to the ACE2. Probably when the immune system is looking for the virus, this is probably existing in this type of conformation. That could be the reason why the virus is escaping from the immune system. So far, we have discussed about how this coronavirus evolved and how it's taken over the body, infecting and causing the disease. Let's discuss about what kind of a treatment and vaccine or cure options we do have. Primarily, we have two different options. One is a preventive measures, which is a vaccine, developing a vaccine. Second thing is treatment options, which are antiviral agents, developing antiviral therapies, antiviral drug molecules. I will start talking about the vaccine first, then we'll go into the treatment options. So, first question raised is, do we have any vaccine or therapy available? Unfortunately, not yet, but there's a lot of hard work is going on to develop the vaccines or therapies. First, let's take a look at how does the vaccine works in the human body. Vaccine is nothing but some sort of a pseudo-virus, a virus-like a particle, looks like a virus, sometimes even a virus that is inactivated. When you inject the virus inside the body, first thing is these antigen-presenting cells will activate and find the molecules, identification molecules, and bring it to the T cells from adaptive immunity. Then T cells generates the memory cells, and B cells generates the neutralizing antibodies. and memory cells remembers the virus, successfully kills, the T cell successfully kills the virus infections when it comes, and these memory cells remembers the signature parts, and then when the virus comes in, they will go and clears them out. There are various different approaches making in virus vaccines. In upcoming slides, I'm going to explain those methods in detail. But first thing we need to know is we need to find a target from the existing virus to use that as a bait in the vaccine to develop the immune system, strengthen the immune system, basically. So in the SARS coronavirus 2, 4 structural proteins are the major sources to develop the vaccine: spike, envelope, matrix, and nucleocapsid protein. First method is virus vaccines, developing vaccines from virus itself. There are 2 different types of methods in the virus vaccines. One is live attenuated virus. That means it's a live virus. It's an existing virus, but it is weakened. It's not as infectious as the real virus. So what people does is they take the real virus, they evolve in the animals and passage for several generations, so that it generates the bad mutations. In general, viruses generates the mutations, bad and good mutations. That's for the viruses in the context of the viruses, not for the humans. When the bad mutation happens, virus becomes less infective. When the good mutations happens in the virus genome, it becomes more and more powerful. So they evolved the live viruses to make the completely inactivated or completely attenuated virus and use them as a vaccine. There are some examples such as the smallpox, MMR, chickenpox, influenza viruses are very well developed using this method. And the second method is using inactivated virus. And again, this is a live virus. It will be inactivated using certain chemicals such as formaldehyde and some sources like heat and evolving in chicken eggs. There are a lot of methods to inactivate the virus. Those can be also used as a vaccine, which will ultimately create the immune response. Second method is using the viral vectors. Viral vectors is a vaccines. Vector means basically you re-engineer the virus, its genome, completely make it inactive or less active using them, and use that other viruses. For example, use the measles virus. And also viral vector vaccines also 2 type methods. One is replicating viral vector. That means it acts like a real virus. It replicates, it goes into the body, makes more of the vaccine. The second type is non-replicating vector. It doesn't replicate. It just goes in there and creates the immune system and sits there. For both types of methods, what we do is you take the RNA or DNA information that encrypts the spike protein and incorporate into this clone into this viral vector and use it as a vaccine. In both methods, that's how the viral vector vaccines used to create the vaccines. And I'm going to have some examples in the upcoming slides on this method as well. Another method is using nucleic acids. This is completely novel, using DNA or RNA as a source of vaccine. For DNA vaccines, usually take the target proteins DNA. For example, for SARS coronavirus 2, most of the people use spike protein genetic information and then inject straight into the body, so that the DNA will be entered into the body and the body generates the spike protein. As soon as the spike protein is generated, the immune system will be reactivated. That's how it works, the DNA vaccines. RNA vaccines, you cannot inject straight. You have to be encapsulated into some sort of source. Maybe it could be lipids or it could be polymers. Then you inject them in, and RNA generates the spike protein inside the body, which will be identified by antigen presenting cells, and immune response will be created. Another type of vaccines is a protein-based vaccine. So in this method, they use proteins, viral proteins, like let's say spike protein. You create the synthesize the spike protein in the lab, purify it, and inject straight into the body. So as soon as the spike protein enters the body, immune system will be responded. And also protein subunit can be used as, for example, I mentioned in the previous slides, this receptor binding domain plays a very crucial part. You can just take that only part and synthesize in the lab, and you can inject them and use them as a potential vaccine. Another method is virus-like particles. So these are also viruses, but entire genetic information is completely removed. These are all prepared completely in the lab, in the test tube. They are artificially, the spikes proteins will be inserted in there by cloning method. And this can be also used as a potential vaccine to create the immune response. So far, from past 6 months, all over the world, using all these various different methods, approximately 141 potential vaccine candidates are identified, and lots of research is going on them right now. Among those 141, 19 candidates, potential vaccine candidates, are already in the human trials, which is a very good sign. If you look at this graph, most of them are coming from either viral vectors or nucleic acids or protein-based vaccines. Very rarely they're using the viruses, live attenuated viruses, and some of the companies are using also virus-like particles as well. Let's take a look at some examples. Before I explain this example, let's take a look at the process, the clinical development process. So when you start working on creating or developing a vaccine, there are multiple stages you have to pass through. Very first stage is preclinical stage, where you develop the vaccine in the lab, test it in the animal, and also sometimes a couple of healthy individuals, you can test them. Once you pass that, you go into the phase 1 trials, where you recruit approximately 20 to 80 people and test the toxicity, safety of the potential candidate. Then it reaches to the next stage, where you go more than 100 people, test the efficiency, how efficiently it is creating the memory cells and neutralizing antibodies. Then it goes to the third phase clinical trials, where you test for the potency. For example, how small amount you want to give to make a big impact. Then it goes into the phase 4 clinical trials, which is basically, in phase 3 clinical trial, you test thousands and thousands of people. Phase 4 clinical trials, basically you release it into the market within a restricted conditions and monitor the patients for long-term side effects. This takes a long time. Long-term side effects means in phase 1 and 2, they will test for weeks, but here it takes months. How patients are behaving, is there any side effects coming or not? This is the whole process. All this process takes approximately 12 to 15 years from creating the vaccine until coming into the market with the FDA approval. But this company, a Chinese company, Consino Biologics, developed adenoviral vector-based vaccine using a spike protein as a genomic source. They use this adenovirus type 5 genetically engineered vector. It's non-replicating viral vector, and they successfully completed the preclinical data. As I mentioned, this preclinical study takes at least a minimum of 5 years to complete, to develop potential drug or potential vaccine. But look at that. They started in December. By February, within 3 months, they completed a preclinical study. That's like a rocket speed. That's an amazing speed. And they also have successfully completed phase 1 safety data in March. So data came out, but unfortunately, 80% of the participants in the phase 1 encountered at least one major side effect, like pain, severe pain, or fever, fatigue, headache, or muscle pain. And also, there's a risk factor with using the adenovirus vectors as a potential vaccine candidates, because from past 35 years, lots of research groups are trying to use this as a potential vaccine candidate, but no success was seen till now. However, this company is still working hard. They went into the phase 2 clinical trials. They're doing some modifications. The results are not yet out. The study is ongoing. So let's hope for the best. with this potential vaccine candidate. There's another viral vector-based vaccine is created by these two companies together in a partnership, ImmunoBio and NantQuest. They are also using same genetically engineered adenovirus 5 vector, but in this genome they are incorporating two coronavirus proteins. One is spike protein and the second one is nucleocapsid protein. Presently, they are in the preclinical stage. The study is ongoing. The results are not out yet. Again, things are going optimistic. Hopefully, we will see probably in 2 or 3 weeks the first stage preclinical data. Based on that, they will move forward with the phase 1 data. Another company, it's a big company, Johnson & Johnson, together with its sister company, Janssen, and American government agency, BARDA. Together, they're spending millions of dollars developing another adenovirus vector-based vaccine, which is 8026. This is another genetically engineered adenovirus vector. By the way, adenovirus is also a virus. It is a virus that causes common cold. It's not that hazardous. And it has been engineered very well, being utilized in the labs all the time for many research purposes. So this potential candidate also presently in preclinical stage. The studies are ongoing. Recently, some data was published, but not quite clear. They are hoping for. They are actually started recruiting for people to go for the phase 1 in July sometime. So they're doing some studies in the preclinical setting. Another company, AstraZeneca, together with Oxford University and American government agency BARDA, they are also using another adenovirus vector-based vaccine to develop the vaccine, an adenovirus vector, this is a little bit different from the other adenovirus vectors. This adenovirus came from chimpanzees, not from the humans. The other ones are the human ones. The chimpanzee adenovirus doesn't infect the humans. This is another good sign. So they have successfully completed the preclinical preclinical studies. They are currently in the phase 1. They're also generated millions of dollars, and they're very optimistic moving forward in the phase 2, 3, 4, and they're working actively hard on this potential candidate, which has some optimistic signs showing. However, again, I mentioned in the first vector, the adenovirus success is mixed. So we cannot anticipate the positive results only. We need to be prepared for the negative results as well. So let's keep the fingers crossed and hope for the best and the vaccine development. Another one, mRNA-based vaccine, which is Moderna Therapeutics here in Cambridge. This company is 2 blocks away from us. They are working hard on developing this mRNA 1273 vaccine. What they are doing is they're taking the mRNA off that encrypts the spike protein, and they are encapsulating that mRNA into lipid nanoparticles and injecting that into the muscles. When it goes inside the muscle cells, it will start producing the spike protein, and then that will be recognized by the immune system to generate the memory cells and neutralizing antibodies. Actually, this one is showing very optimistic results. They are also working on a rocket speed. Like preclinical data was completed within 3 months, very optimistic results obtained. Phase 1 completed within 2 months, very optimistic data obtained, it passed complete safety tests. Currently, they're doing the phase 2 studies, which will be, the results may come out in a couple of weeks by end of June. Actually, they're planning for a phase 3 already, recruiting approximately 30,000 people to go move forward. This vaccine is showing some very positive, optimistic signs, hoping for the best. So far we have discussed about the potential vaccines and their development. Now let's talk about the therapeutic options, such as antiviral agents and drugs. There are 2 methods are being used to develop. The first one is repurposing of the existing therapeutics. Using currently available drugs, try them on the coronavirus. That's the first method. Second method is based on the genomic information of the coronavirus, develop from the scratch, start from the basic research. The first one is much faster than the second one. And in the first one also, there are two different approaches. Number one is first use the broad spectrum of antiviral agents that are already approved for various different viruses. And the second one is all other potential drug candidates approved for various different diseases such as blood pressure, fever, heart diseases, and so on. So far, all over the world, many research labs, many companies collaboratively working on developing therapeutic targets, therapeutic agents. Approximately 249 potential molecules are under development. Out of 249, 176 are already in human clinical trials, which is very optimistic sign, very hopeful sign. Now I'm going to show you some repurposed drug molecules. So these are all the antiviral agents that are approved except this molecule, remdesivir. So the chloroquine, this is a treatment for malaria. When it was tested against coronavirus, it showed some signs, and when tested for which target it is targeting, the clear data was not available, but they assume that it's targeting multiple proteins in the coronavirus. These two are protease inhibitors that are approved for HIV, successfully working for the HIV, and they also show some positive vibes towards the coronavirus. This one developed for Ebola virus, and it is targeting the spike protein in coronavirus with some positive results. This one is also developed for hepatitis C virus. It's also showing some activity towards the nucleocapsid protein coronavirus. These three are targeting the replicase, the RNA-dependent RNA polymerase. These two are approved drugs for Ebola and hepatitis C and influenza as well. But this one is actually under development. Gilead Life Sciences started working on developing this particular drug molecules targeting the Ebola virus. While this work is ongoing, the COVID crisis came in and they tested this, repurposed this towards the coronavirus, and then which showed very positive results. The other ones, all these other ones, even though they showed some positive data, but when the phase 2 clinical trials, the efficiency was not as great as the remdesivir. Let's take a look at the remdesivir. What it does is it basically blocks the replicase activity to stop the viral infection. And this is the structure of the prodrug. When it enters into the body, it becomes the active drug like this, which is a mimic of adenosine, a nucleotide that makes the DNA or RNA genes. This remdesivir is a crystal structure. This dark blue one is the virus RNA, and this is a template where the RNA polymerase started making the new viral RNA. The mechanism is not quite known, but the binding properties are shown. The drug molecule goes and binds to the replicase molecule, which is a positive sign. So this molecule also successfully completed preclinical phase 1, 2, 3. All the studies are completed. Now it is in the phase 4 study. And also this drug molecule, FDA approved in the US for the emergency use in required conditions by taking the patient's permission. Another interesting molecule also, EIDD-281. This is also a potential drug candidate that was targeted to develop Ebola as a potential candidate for the Ebola, which also blocks the replicase activity. This was developed by Emory University together with Ridgeback Bio and Merck Pharmaceuticals. This is the structure of prodrug structure of the EIDD-2801, and it also mimics one of the bases in the DNA bases, uridine. This molecule is a little slower than the remdesivir because this was in early stage. Preclinical data is completely successfully completed. Now it's in the phase 1 which is also going well. And there are some lots of hopes and positive, optimistic hopes are there for this candidate. And one more interesting property of this particular candidate is it can be taken like a tablet orally, like a pill, but remdesivir has to be administered like an injection or IV. There are some other type of therapies also developing. This is what we are doing in my lab. Unfortunately, I cannot show you some data because of the confidentiality and conflict of interest issues. But I will give you a glimpse of what we are doing in my lab. So we are isolating antibodies, human antibodies, the developed in the human bodies, and testing them against the SARS coronavirus. If they are working, if they are not working, then we are modifying them using some computational algorithms, and then go back in the lab and synthesize them and test them again against the coronavirus. So this is a crystal structure that published recently. This is a receptor binding domain of coronavirus, like this one here, this is a ACE2 receptor. This is how it binds, and this is a human antibody, which also binds to the receptor binding domain, but not exactly where it's supposed to bind. That's why this is not very active, or this is not useful. That's why we take these type of antibodies and then we re-engineer them and we resynthesize them and try them to test against the coronavirus. This is a nucleic acid, protein sequencing map of the SARS of this spike protein, which has two subunits, S1 and S2. In the S1 subunit, which has an RBD domain that binds to the ACE2, S2 subunit helps the virus fusing into the cell.
You think?
So far, I have covered the evolution of the coronaviruses towards the COVID-19 pandemic. Then I have discussed about the immunity body surveillance system and its crucial role fighting the viral infections, and how its failure leads towards infections and diseases. Then I also have discussed about the potential therapeutic and vaccine development options. However, there are three different types of pathogenic coronaviruses that I discussed in the beginning. The question is, do we have, we already knew that SARS coronavirus 2, there is no vaccine available still. But is there any vaccine available for MERS or SARS 1? Unfortunately, not. They are there since 18 years. Approximately SARS 1 is from 18 years and MERS is about 8 years. Lots of research is going on. Still, that we are trying to find a potential vaccine candidate for these also. Another way we are trying to do is a pan-coronavirus vaccine. One vaccine can act all sorts of coronaviruses. That's also lots of research is going on in that sense. So since there is no vaccine, what are the options we have to survive? The most important thing is we have to live like virus is with us at this current situation. This is mostly a chemist's perspective of explaining the current social distancing and using personal protection equipment. These cartoons are lots of chemistry in there. First of all, this picture, I would like to hear thoughts from your students. This is a chemistry. So I'll explain to the general audience later in a minute, but I would like to hear your students' thoughts from your college. Take a look at this. Please post your comments. I'll come back to it later. But this is a recent data published about social distancing and using masks. Which one is better? Which one successfully protects from the infection? The data suggests that this data is analyzed from the three different countries, United States, Italy, and China, where the more infections are evolved. The data suggested that both are very important. Social distancing as well as covering the face mask. Both of them are very, very important to stop the infections, spread the virus. And this is, again, a typical chemist's perspective, homo-lumo molecular orbital theory. But I'm using this, staying home is very important. Unless until it's necessary, don't go outside. Staying home, protect yourself. This is heating healthy. Explains the importance of eating healthy. A fluorine molecule eating the electrons. Fluorine is always electronegative and it's hunger towards the electrons. Get some rest. This is melatonin, sleeping. That's another symbolic, a cartoonistic expression. Stretch yourself, do some live active lifestyle, do some exercise. When you shine IR light on an aromatic molecule, it becomes so highly hyperactive. That's what this says. So all this helps your adaptive immune system to be strengthened up. And now, here in this cartoon picture, one of the carbons wearing the mask, but the other one is not. So this is yelling at the other carbon saying, "Where is your mask?" Saying the importance of wearing the mask. And also, if you look at the Newman projection formula, this is syn confirmation, right? Both hydrogens are kind of close distance, but when they talk to each other and this hydrogen is not wearing the mask, once this carbon got the mask and rotated 180 degrees centigrade, which becomes an anti-conformation where all the molecules are kind of away from each other, these two hydrogens and even those two hydrogens. That indicates the importance of social distancing, using personal protection equipment, and also following the personal hygiene using the sanitizers and etc. So in this my journey, there's a lot of people involved, lots of my colleagues from all these prestigious institutions, my colleagues, my students, my mentors. My chemistry life journey started from Department of Chemistry, OU campus, Usmania University, where I started learning chemistry. Then I did a little bit of research, CSI, IICT. I didn't stay long over there, but I set up my first reaction over there. I still remember that the experience. It's very outstanding colleagues. Then University of South Carolina Chemistry Department, I got my PhD where I was trained as a pure organic chemist. Then moved to Wayne State University as a postdoctoral scholar where I learned molecular biology, working with bacteria, cloning the bacteria while I'm making compounds, synthesizing compounds using my organic skill set. Then I moved to my Massachusetts Institute of Technology, MIT, where I started learning working with the mammalian cells, cloning, studying the biosynthetic pathways, making the viral vectors, while I'm using my other skills like organic synthesis. Now, at Expansion Technologies, I'm working with human organs, human tissue sections to design, develop early diagnostic methods for various different cancers so that we can save many lives. I would like to thank all the colleagues and students and mentors for their support, encouragement in this journey. Last but not least, I would like to thank my family and my friends for their unconditional support in my success and encouragement, support and encouragement. Without their encouragement and support, I don't think I would have been where I am and what I am today. Having said that, I think my apologies, I took 10 minutes more. Thank you so very much for your attention. I'm happy to answer any questions you have. Thank you. Thank you so much.
Yeah, sir. Thank you for an excellent presentation, sir. Uh, Thank you for an excellent presentation, and the response is overwhelming. Okay. Uh, Hope are you able to hear me, sir? Mahender Garu?
Yes. Thank you so much.
Very,
Looking forward to the questions.
yes, sir. Dr. Ishwari, please unmute yourself and please ask the questions whatever there in the chat box.
Thank you, sir. Dr. Manjusha ma'am asked about what is the lifespan of a coronavirus inside the human body.
That's a great question. As long as again I said, the viruses are not live animals or live cells. As soon as it releases the RNA inside the human cell, it disassembles. All it does is hijacks the mechanisms of the human cells. So basically, there's no life of virus. It's all about the nucleic acid RNA, which human cell enzymes makes more and more and creates more viruses. I hope I answered the question.
Thank you, sir. Afshan and Shweta madam asked about will the same person get COVID twice?
Yes, that is possible. In fact, there are many patients It's like this. Again, this is a completely new virus. Within 6 months, it's only 6 months old. There's a lot of unknown things that we need to know. A month later, coming back for the test is positive. These kind of patients, there are not a lot of them, but there are some of them there. So absolutely, there's an absolutely chance of reinfecting. It's a virus, you know. As soon as your immune system goes down, it attacks you.
Sir, can a patient transmit disease during incubation period?
Absolutely, 100%. And he doesn't even know that he is transmitting, because this virus has two stages. First thing is, as soon as it gets into the nose or mouth, through the mouth, it starts replicating, making more and more in the upper respiratory tract, from throat to the nose. This is called upper respiratory tract. And gradually, within these 2 weeks period of time, it goes down towards the lungs, which is called lower respiratory tract. That's when it enters, it becomes more and more severe. But when it is already more down here, when you sneeze or cough or even speak loudly, it's going to come out. It travels 50 miles per hour in the air.
Thank you. Thank you, sir.
Sir, could you please any more extra precautions to old age people?
The most important thing for the old people is, strengthening the immune system, active lifestyle, which is one of the, walking activity, some sort of activity they have to do is the one is important, and eating healthy diet, healthy food. That's very important to strengthen the adaptive immune system. Sometimes immune supplements also helps to strengthen for them. Other than that, stay away from the COVID suspected patients or anybody. Like, don't go outside in this current situation. For older people, I am always suggesting that don't go outside unless until it is necessary. Even people who are going outside, they have to consider themselves as transmitters. For example, even though they are not infected, but they may be contaminated. Their clothes or their body may be contaminated with the virus from the air. As soon as they comes home, they have to take precautions like, go straight to the bathroom and then clean them up themselves before they meet their near and dear.
One more question, sir. Could you please explain about cleaning of vegetables, green leafy vegetables and fruits?
Yeah, that's a very good question. Lots of people are asking me about it. So it's difficult. These are the fruits and vegetables, right? What I'm trying to do is I buy rubbing alcohol from a pharmacy, which is a 70% ethanol or isopropyl alcohol. Whatever I bring home, I spray them on it and wipe them using a paper towel or a cloth. Then, after 5 minutes, I wash it with water and then rub it again and put it in the fridge or eat. That's one of the things that I do it. The rubbing alcohol, basically, the study says that within 5 minutes it kills the virus. Rubbing, which is nothing but a disinfectant, which is a sanitizer. But don't use the bleach or Dettol or anything. No, that's not recommended at all.
One more question, sir.
Sure.
How can we trust N95 mask or a cloth mask which we made in home?
Something is better than nothing. So any protection, it doesn't matter. N95s are very well tested in the laboratory. All the time hospitals are being used. Those are filters, I think, nanometer particles successfully. Micrometer or nanometer particles successfully. N95s are very well tested. But again, a thick cloth also we don't have any scientific data, but lots of even doctors and scientists are recommending having something is better than nothing. So protect yourself by covering them with any cloth or mask or whatever it is.
The mask which we buy outside, shall we wash once or twice while using?
What type of masks are those? Use and throw masks or a cloth mask or what it is?
Thin cloth masks.
Thin cloth masks, right? Yes, you can wash them. And every day I would recommend, as soon as you come home, put it in soap water for 5 minutes and then you're ready to go again for the next time.
Yes.
What about
And
N95? N95 mask.
N95's reusability is little hard. Yeah, there are some stations, you have to disinfect either using UV light or put it in the cooker. Pressure cooker is another big disinfectant. Like once it goes up to 120 degrees, that's what we use. Sterilize materials in the lab.
Sir, one question here, sir. Can our body
Yeah,
differentiate between the bacteria and virus, sir?
of course. Yes. I mean, even virus between virus also, different virus also can be distinguished.
One more question, sir. It maybe sound like a Trump-like question. When we can able to disinfect the virus using a soap water when it is outside our body,
Yes.
uh, why can't we do some kind of a mechanism which is inside the body? It is a layman question maybe, but we need to answer it, sir.
Yeah, it's see, for example, let me give you an example. Bleach. Bleach contains chlorine. Chlorine is highly poisonous. Somebody asked the same question about the bleach. Can we drink the bleach? No, not at all. It's just chlorine. Why can't we drink? It's poisonous. Something goes inside the body. Again, it's a chemical. That's why it's taking 12, 15 years to develop a small drug molecule, a vaccine, right? If it is that easy, and you could have had drug molecules or vaccines already, right? So poison is a poison inside the body. Outside, there's a difference between inside the body versus outside the body. Soap water, yes, outside the body it's fine, but inside the body there's lots of sensitive cellular mechanisms present which may harm. Again, soap is basically a detergent, right? It disrupts the cellular membranes. So virus membrane, it goes into the virus membrane and breaks into pieces. Same thing in the cells. Inside human body, we have cells. It goes and disrupts the cells, and then again, it starts to become toxic. That's why.
Thank you, sir. One more question here, sir. Is
Sure.
eating habits of the persons is related to the viral infection, sir? Because everybody is blaming the Chinese meat market for it. Is there any relation between the consumption of the meat or eating habits to the coronavirus infection?
The answer is yes and no, because it's not. I wouldn't call this an eating habit. Eating what you are. I mean, be careful with what you are eating. Let's say some people eat meat, right? Of course, the virus stability on a meat is much stronger than any other surfaces. So if you clean it well, that should be fine. I mean, especially in Indian cooking, we boil the food, so virus is already dead. Not to worry about. As long as you're not eating the raw meat or raw food.
Okay, thank you, sir. So one more question regarding the plasma therapy, sir.
Please go
So
ahead.
yeah, plasma contains the antibodies of the coronavirus. What?
Neutralizing antibodies.
Yeah,
Yes.
yes, sir. Is how effective it is the plasma therapy, sir, and is it advisable? Lot of discussion is going on. Please throw some
Yes.
light on it,
Yes.
sir.
That's a very excellent question, Andy. So when virus infects, each individual's body responds its own unique way, and it generates neutralizing antibodies. As I mentioned in my talk, the B cells creates the neutralizing antibodies, and those antibodies varies from person to person, region to region. Person in America has a different type of antibodies. Person in India may have a different type of antibodies. So you have a different type of antibodies, and Ishwari may generate different type of antibodies. So they are very vast different. But out of the, like, let's say 100,000 antibodies generated by the body, some of them are active. So we don't know which one are active. That's why they are using entire plasma. It's a trial and error thing. But some of the patients are showing recovery, which is a good sign. But that's a good thing.
Yeah, sir. One more question in the chat box is that
Please go ahead.
taking the citrus fruits and other things, will it help in maybe preventing or in the cure of the coronavirus? And what is the basis for it, sir?
Eating fruits, vegetables, all this stuff helps to the body to strengthen the immune system, not to kill the virus. That's one important point you need to keep in mind. That's why I mentioned healthy diet. Eating all sorts of citrus fruits basically gives the vitamin C to the body. So regular fruit juices, yes, good for health. fibrous food, good for health. All those, vegetables, carrots, all those things. Carrots give the vitamin A, and beans gives the vitamin B. Lots of things. So these are all going to help the body to develop a stronger immune system. They're not going to go and fight with the virus. Just citrus food is not going to do anything to the virus.
Thank you, sir. One more question here, sir.
Go ahead.
What is the origin of this virus? If somebody says that it has generated in the meat market was there from years together, and people are consuming different kind of meat, maybe in the China or anywhere. Suddenly, from where it has come, and I want you to throw that actually how the virus is generated in general and specific to this coronavirus.
It's another very good question, Andy. So, viruses are evolved. They evolve in the body. So, in my previous slides, I mentioned that the coronavirus is originated from bats. Somehow, how not clearly known, somehow it is transmitted into intermediate hosts like pangolins, civet cats, camels, raccoon dogs, all these type of various animals. And now you said it came from the Chinese meat market, right? In China, lots of people eat various different types of animals. Somehow, from forest, from bats may have had some kind of interaction with these pangolins or something. And the pangolins are the reservoirs of the virus. They don't show any symptoms, but they harbor the virus. When you eat it without cleaning well, most of the times Chinese eat raw meats a lot. They don't cook very well as we cook. So that could be the reason that came from bats to cats, camels to the humans. Again, I say this is a zoonotic. It can infect animals, it can infect humans, but it exists only animals.
It's one more question, sir. Is it an intelligent virus?
Very intelligent, smart virus. It's not about the intelligence. Viruses, always very, very clever objects, very smart. They hijack enter this big human body, huge human body. It's a small 20, 50, 100 nanometer particle shakes the 150 kilo human, right? So how smart it could be.
Yeah, one more question, sir. What is the chances of getting the virus from the mother to baby when she delivers?
Very rare, Andy. Very low. The HIV type of virus la kakunda, some rare cases koni kanapade gani, most of them not quite.
Thank you, sir. Any concerns from Ishwari from your side?
Sir, at what temperature virus can be killed? Are there any studies about temperature and
Yes.
type of virus?
Yes. I have created a nice table. I will forward it into your WhatsApp group. I showed that in my 5th slide, actually. The virus is stable, very stable at 4 degrees centigrade in the fridge for up to 2 weeks. And it is stable for 2 days at body temperature, like, in the sun, 37 degrees centigrade, up to 2 days. But it is dead at 70 degrees centigrade in hot water. This virus, I shouldn't call a word called dead, but it will be cleared, eradicated at 70 degrees centigrade within 5 minutes.
Okay. Any more questions, Amma? Vanaja from Ishwari?
Sir, one more question.
Please.
Heard that vegetables cleaned with baking soda and warm water, is it going to kill the virus? Is it?
The answer is I don't know, but I can interpret the concept. Because I have never done that. There is no data available to say it does happen. It is good. I don't know the answer because no scientific studies were conducted on that. Let's say baking soda. What is it? Baking soda is sodium bicarbonate, right? Sodium bicarbonate is a salt. It's so a salt creates osmotic pressure, right? Osmosis. So probably it creates osmosis in viral membrane and it may kills, but I don't have any data to say it does. So I don't know the answer for that. But just as a scientist, I'm interpreting why people are using it. Hot water, again, I said, don't use the boiled hot water on the vegetables; they go bad. Best method is rubbing alcohol, which is 70% ethanol or isopropyl alcohol.
Thank you, sir. Sir,
Is
please go ahead.
is our pet animals are safe, sir, in this time, Corona time?
What sense?
For
Do
spreading
they get infected?
Corona.
Do you mean they get infected?
Yeah, yeah.
There's a possibility, of course. It's an animal virus. It transmits animal to humans, human to animals. There is a possibility, but I haven't seen any instances so far.
Any more questions, Amma? And sir, please, Ishwari.
Subara from me. One question from me.
Yeah, please, ma'am.
Yes, yes, please.
So I heard that people with O blood group are less affected by coronavirus than the other blood groups. Is it true?
Yes, that's true. There is a couple of studies, not a lot of studies were done, but some good significant data is showed that the O plastic group blood group people are kind of more resistant to this virus, probably because their body generating very efficient neutralizing antibodies. I mean, see, immune system is nothing but white blood cells, right? All the white blood cells are the immune system. So possibly, that's a possibility. Yes, there is some data available on that.
Thank you, sir.
Sir,
You're welcome. My pleasure.
please,
Sir, one more.
Yes, go ahead. Take your time, no problem.
Sir, what is the status of the whether it is a drug or a vaccine or antibodies? How much time it takes to come into the market, approximately, based on the status?
You didn't pay attention to my talk, probably. No, I'm just kidding. I'm just joking. So, last 5 slides. See, vaccine and drug development. I'm anticipating the therapeutic targets. Probably end of this year. I'm hoping for remdesivir or ED 2801. Probably be ready for some sort of a emergency use, emergency treatment. But for the long-term side effects, we need more time, like a couple of years. And vaccine, the Moderna's mRNA therapeutic is showing very promising progress compared to other, I don't know, viral vector vaccines. I'm looking forward to it. Fingers crossed now. It may complete the successful study by December, but still again, approval may take a year. But it may be ready for emergency use, which will be protected many more people, I guess.
Sir, please throw some light on herd immunity, sir. How it works and herd immunity, how it works, and
Herd immunity, what is that?
that is, I don't know, if many more number of people get infected, there is less chance that other people will get infected again and again, something like that.
I never heard of that. I don't know. I'm not sure about the herd immunity.
Any more questions, Amma? Ishwari? Avanaja?
No, sir. Thank you.
Sir, very excellent presentation and followed by very good number of questions and your answers, sir. I hope that all the
Thank you.
participants are satisfied with this. And most of the times, what is happening, sir? We're all WhatsApp graduates. So the knowledge, whatever we got from about the coronavirus, is through WhatsApp groups, some fake and some original.
Be careful. I would say be careful with
Yeah,
that.
that is what. So,
Very, very be careful.
okay, yeah, it is a very excellent and in-depth presentation for about the coronavirus, and all those who are chemist students and all those who are not chemist and belongs to other groups also, they have responded very well in the WhatsApp chat, and we are happy that we had this webinar, sir. Now I request Ishwari to propose the vote of thanks.
Before that, I have a small question for you all guys.
Yes.
Did any student respond to my question on my last slide? Any comment? I'm curious to see.
Yeah. Okay, sir. No, sir, I haven't seen anything.
Yeah, don't worry about it. We will talk about that offline. Go ahead. It's getting late already, probably.
Please,
Thank you, sir. Dear participants, thank you is the best prayer that anyone could say, quoted by Alice Walker. So, on behalf of Department of Chemistry, Sarojini Naidu Vanita Mahavidyalaya, I feel honored to propose a formal vote of thanks. I would like to express my sincere gratitude to our speaker, a very accomplished scientist, Dr. Mahender Dewal, principal scientist and founding member, head of research and development, expansion technologies, Cambridge, MIT affiliate, USA, for kindly accepting our invitation in spite of his hectic schedule and different time zone to deliver a talk on COVID-19, a pandemic of coronavirus, underlying chemistry from cause to cure. I am proud to mention here that I was fortunate to be his classmate in PG, Department of Chemistry, OU campus. It was indeed a very informative, motivating, and knowledgeable session for both organizers and participants. Let me acknowledge him for sharing his immense scientific knowledge and technical proficiency on the evolution of coronavirus towards COVID-19 pandemic, therapeutic and vaccine development process, body immunity system for fighting virus infections, self-protection measures, and various treatment options. I am very glad and thankful to all the participants for their excited participation and their cooperation. Especially, I appreciate the presence of many overseas participants. Thank you for joining us. I hope this webinar was beneficial to update their knowledge. I would like to acknowledge my sincere gratitude to our management for their constant support. A heartfelt gratitude to our motivating principal, Dr. Shobana Madam, for encouraging us in all our endeavors. Behind every successful program lies the manifold effort of committed faculty. Let me mention my special thanks to our retired chemistry faculty members and Dr. P. Saroja Madam, former principal, for guiding us in all aspects during the process of our webinar. I wish to thank my colleagues from the Department of Chemistry, all the faculty members, friends who have always encouraged us. I also would like to sincerely appreciate the technical team comprised of Mr. Subbarao sir, HOD Physics and Electronics, for his technical assistance. Finally, a big thank you to our distinguished speaker and each one of you for being with us. Thank you one and all for making this webinar possible. Thank you, sir.
Thank you, Ishwari. Sir, if anything is to say, sir, Mahender Garu?
Thank you so much for inviting me
Yeah.
to give this lecture. I appreciate it. It's my pleasure to be with, even though it is virtually, with you all guys, with your students. Thank you. Thank you so much.
Ma'am, with your permission, I will try to end of the session now.
Thank you.
Yes, Subbarao, you can go ahead.